First, we're going to have a talk by Professor Adriana Tomic. She is a brand new assistant professor who comes to us from the UK. She has a joint appointment at the NEEDL, which is the National Emerging Infectious Disease Laboratory here at Boston University, one of the only BSL 3 and 4 rated facilities in the United States. And she also has an appointment in biomedical engineering as well. And so that makes her a very unique person at BU. She's a computational scientist as well as a person looking at using wet lab data to do her work. So her work requires a lot of experiments. So that makes it a really nice match with damp labs. So I'm pleased to welcome Dr. Tomic.
Thank you, Katie, for a nice introduction. And thank you, Damp Lab, actually, for this opportunity to develop this assay and to present our preliminary data today. So the assay I'll be talking about is a high-throughput platform for analysis of adaptive immune responses, or we term it as Systems Cytomics. So my lab, in our lab, we are interested in understanding one of the most remarkable features of immune system, and that is the ability to remember. That notion that immune system remembers we have a written proof about it some 2,400 years ago when Greek philosopher Thucydides wrote about the Athens outbreak of plague in Athens and noted that the same man was never attacked twice, never at least fatally. So for how long immune system remembers? Well, it turns out it can remember for quite a long time. Majority of you had chickenpox when you were a baby, and you had it once and never again. On other aspect, we can also induce some immunity, which can last even for a lifetime. And we were very successful in generating vaccines that can induce such immunity, such as, for example, very successful story behind the poliovirus vaccine, which almost led to complete eradication of this virus around the globe. Some other examples are also vaccines against yellow fever virus or measles. You get this vaccine once and you are protected for a lifetime almost. But if our immune system can remember for quite a long time different pathogens and provide protection, then why are some people more protected than the others? So this is kind of a billion-dollar question, and we immunologists, clinicians, biomedical researchers are trying to better understand differences between individuals and in their levels of protection. So in recent years, with this boom of omics technologies, this kind of brought kind of holy grail, and we expected that this will provide an answer to that billion-dollar question with identifying correlates of protection. So identifying genes, transcripts, or proteins which are specifically associated with protection in some individuals against some diseases. But on one hand, when we think about these omics approaches that we currently have, genomics tells us what can happen, transcriptomics what appears to be happening, and proteomics what makes things happen. So none of these approaches actually tell us what is happening now. So I think that we need to rethink our approach in defining correlates of protection and start thinking about the smallest and the simplest unit of our biological system, and that is a cell. So cells actually give rise to a process, and we need approach like a cytomics approach in which we can get high throughput information on the cell level. So for example, for immunity, we know that immune cells have a capability to produce proteins which kill infected cells. So you see here the smaller cells running around are human immune T cells, and they recognize these infected cells in red and kill those cells. So this process, just killing of infected cells, gives rise to immunity, protection of against the disease. So the proper question we should be asking here is, why are some immune cells more protective than the others? So when we turn this question around, now we can try to tease out the cell-mediated correlate of protection. So since the time of Thucydides, today, 2,400 years later, we a little bit better understand the process behind this immune memory. And we know that adaptive immune system is the one that carries this memory through memory B and T cells. So these cells, memory B and T cells, are generated upon exposure to an antigen, to a virus, and these cells then remain in our bodies and can provide protection if the same virus comes again. These cells expand very, very quickly. They can quickly kill the virus, and you will have a less severe disease, less symptomatic disease, and so on. So adaptive immune system has ability to record every immune event over your lifetime. And we can then tease into this specific adaptive immunity arm and look into T B cells to better understand your past exposures, and then use that to generate better vaccines and protection against different diseases. So B and T cells are lifelong guardians of our immune system, and this is just a very quick kind of basic introduction into immunology. So very simplified, B cells have the ability to produce antibodies, and these antibodies can recognize the virus, code the virus, prevent virus from entering and infecting cells. But T cells, on the other side, so killer T cells, they can directly recognize infected cell, kill that cell, as I showed in that video, previous slide. So these two cell types complement each other, and it turns out that T cells actually can provide protection in the case when antibodies are still not generated, and that is the case with all the newly emerging pathogens that are showing up, to which we don't have immunity to. So this is a diagram from a deadly strain of avian flu. So avian flu, which has 50% mortality rate. So in comparison to SARS-CoV-2, if this strain would to spread around, that would be disastrous. So in this case, these are two diagrams showing two different onsets of diseases. And we see the case when patient recovered from this flu virus, T cells emerge, killer T cells emerge 10 days after symptom onset, preceding what we see antibodies, which took around 2 weeks to develop. So T cells, this window here, when T cells appear, they can control the virus, reducing viral infection, viral load, and help antibody generation here. On the other side, this is a patient who died, unfortunately, due to this virus. We didn't see any T cell appearing, and antibodies just took too long to appear, I guess. So on another note, similar thing was noted with SARS-CoV-2, and T cells were associated again with less severe disease. To see here in blue, they preceded antibodies again. And in the case where you have a severe SARS-CoV-2 infection, T cells were absent. They did not develop early in infection. And also our data show that T cells that are generated very, very early after infection are associated with having a long-lasting and durable protection up to 6 months after infection. So clearly, CD8 T cells or killer T cells are very important to provide the protection against newly emerging viruses with pandemic potential, and that's why we are interested specifically in that population. Even though these cells were recognized very early back in the 50s, 60s, with their ability to kill tumor-infected cells, that's why they were called killer T cells. But nowadays, we know that these cells are much more than that. So they have ability to modulate immune responses by production of different cytokines. And here just showing, again, very simplified, these T cells upon activation, they can produce different types of cytokines that can activate cellular immunity, activating macrophages, dendritic cells, neutrophils. Type 2 cells can, they induce production, activation of humoral immunity, antibody production. Type 3 mucosal immunity, and then they can also provide immunosuppressive functions through regulatory production of regulatory cytokines. Also, they can establish a lifelong memory. So this is a newly described subset of CD8 T cells, which is formed early during the expansion of these cells. And these cells, they seem like stem cell-like cells. They have a self-renewing capacity, and they were so far measured to last at least 30 years after infection or vaccination. We can still detect them in our immune system. So in order to reveal the full potential, full spectrum of human T cells, we need a better high-throughput assay to do that. First of all, a lot of times we immunologists use antigen stimulation assay. This tells us what these T cells recognize. So you can put different antigens here and then just looking at activation of cells. For example, this is from our work where we look at T cell responses to different SARS-CoV-2 peptides and their interferon gamma production using ELISpot, so ELISA-based assay. ELISA-based assay, what it does, it captures just the interferon gamma production. But as I described, like all these white wells here basically tell you these cells do not produce interferon gamma, but it doesn't mean that they do not recognize this peptide because they might be producing something else. So we need to improve on that. So ELISpot is one-dimensional. On the other side, we have in recent years, in the last decade or so, cytometry-based assays were improved in so much that we can now do, we can stain with at least 40 different antibodies, 40 different proteins analyzed on different cells and get information on about 100 or more different cell phenotypes and subsets using mass and spectral cytometry. So with these approaches, we can then get information also how cells react, which cytokines they produce, how they look like, and so on. So you can get a full in-depth characterization of T cell signatures and different flavors. So the idea was to combine then these two assays into one high throughput using a pipetting robot here that could facilitate preparation of simulation assays in 96 well plate here, and then also staining for this high dimensional cytometry. And this is the Systems Cytomics assay that we promote. So luckily for us, Dump Lab decided to support this assay, and all this work wouldn't be possible without your support and help on this. So for the Systems Cytomics, what we did is in this 96 well format, currently we can put 12 different conditions and test immediately 8 different donors here. And then we developed a spectral cytometry 39 antibody panel, which can allow us to look at all these different types of cells, different cytokines produced, and so on, as I described. So we can look at polyfunctionality. What else we can do with this assay is look at the cross-reactivity. So within one individual, we can say if these T cells from that individual can recognize different flu strains or different SARS-CoV-2 coronaviruses, which we put in our assay here. And also we can determine the phenotype. Are these cells forming as long-lived memory CD8 T cells? Are they exhausted? How they look like, and so on. With this, having covering the phenotype function, specificity, and clonality of T cells, we now can start our quest for this T cell-based immune correlate of protection. So I'll give you a brief run through the results of our assay and things we discovered. So we put different antigens from seasonal and pandemic flu and coronaviruses here. And I can show you here, this is the clonality, so antigen-specific expansion of cells across these 8 different donors. So in red and orange are different flu strains, and in green we have different coronaviruses, including SARS-CoV-2 as well. So we can see that around 3% of cells in your circulation are recognizing pandemic H1N1 flu virus, as well as SARS-CoV-2, so quite high percentage of the cells can be detected in blood. So we can also describe these cells in terms of immunity that they stimulate. So we see, for example, for this seasonal coronavirus, OC43, that these T cells rather produce very strong interferon gamma responses, driving cellular immunity, as well humoral immunity, which then we see that humoral immunity is rather with SARS-CoV-2, after stimulation with SARS-CoV-2, rather induced. And we can also look at all other cytokine responses as well. Next, we can then evaluate distinct cell signatures across different viruses. So for example, this is a seasonal flu virus that is constantly in circulation. And we see that T cells are rather inducing this mucosal immunity, which gives a kind of a protection in the nose where you're constantly exposed to this virus, and therefore you can be protected against that virus. On the other hand, if we look at this is a pandemic H1N1 virus that we had, we see that there is also strong induction of cellular immunity as well. Similar signature was observed for coronaviruses, so seasonal coronavirus, very, very strong cellular immunity here induced. And for SARS-CoV-2, it seems like T cells are still not deciding in which direction and what is a proper immune response to tailor to this virus, because we see equal distribution of an induction of different kind of immunities by T cells here. We can also identify donor-specific T cell signatures. For example, you see these donors 5 and 6. They have quite cross-reactive potential, and their T cells can recognize all these different strains. So this is also something to look for. So with all of this, I think that Systems Cytomics and similar approaches can lead toward precision vaccinology, which is kind of an overarching goal of our lab, to tailor vaccine strategies and vaccine candidates for individuals, depending on your immune history and on your T cells, let's say. So yeah, with this, I would like to, so the antigen stimulation assay was developed in collaboration with Human Immune Monitoring Center at Stanford, Mark Davis and Holden Macker. Then I had incredible support here at BU for development of high-dimensional cytometry assay from Anna Balkina at our amazing BU cytometry core facility, and Jen from microbiology department for her advices on development of this 39 antibody panel. Of course, I'm very grateful to people in my lab, Ivan and Matt, who will now take over further developing this assay, especially since we now obtain our own OT2 last week. So very looking very much forward to establishing this assay within the needle. And as I said, this work wouldn't be possible without DAMP Lab and the entire team. So Katie, Doug, and Courtney. But biggest thank you goes to lady boss of the OT2 OpenTron, Rita Chen, and she did an amazing job. And all this work here wouldn't be possible without her. So thank you very much. And yeah, we are recruiting. So if I'll take this as an opportunity to spread the word. So thank you very much.
Can you tell me about the donors? Are they healthy when they donate for this particular?
Yeah, so these are healthy donors that are donating basically blood. So yeah, so the first idea was just to establish to run the assay and have it. But then we have a huge cohort of SARS-CoV-2 infected people that were followed for 2 years during vaccinations, having Omicron infections, breakthrough infections. And the idea is then to apply a similar approach there to better understand what is happening on the T cells.
Okay, so you would be able to put the clinical background.
Exactly.